Okay, so this is our class. So sorry, I should have deleted the title here. So this year we are only having one course number, Stats M254. And if you know, like this is a class, sorry, this is a core course designed for PhD students in bioinformatics. So if you know that some of your friends still have enrollment issues, you can ask them to email me and we'll try to accommodate everyone's enrollment. And so the class time is a little bit tricky because we originally had our class scheduled at 3:30 on Mondays and Wednesdays, but then I was told that there would be a bioinformatics seminar at 4 p.m. every week. So this quarter they moved it back to 4 p.m. instead of the 11 a.m. So we have to avoid overlap. So that's why for now the class time on Monday is at 2 p.m. But it's a little bit inconvenient for me because I teach another class, an undergrad class that's 100B from 12:30 to 1:45. So that only gave me like 15 minutes in the middle. So I'm trying to ask people here if we can start this class at 2:30 on Monday so that we can start 2:30 and finish at 3:45. Would that work for everyone? So if so, I think that can give you a longer break between the two classes and will be better for me. But if that doesn't work for you, you can let me know. So if 2:30 works for you, so I'm trying to ask if 2:30 starting at 2:30 will work. If it doesn't, can you raise your hand? I see one thumb up. That means in support. And how about Wednesday? Will Wednesday 2:30 work for everyone as well? If so, maybe we can just start both Monday and Wednesday lectures at 2:30 to avoid any confusion. Because at least I think for now, the first 2 weeks will be remote, so we don't have any classroom issue for that. So if starting at 2:30 will work well for people here, I will make an announcement after today's class. At least we'll do this for the first 2 weeks. Thank you. Okay, so this is the class called Statistical Methods in Computational Biology. So the goal here is to build a very, I would say, straightforward bridge between statistical methods and bioinformatics applications. So our goal is to introduce some cutting-edge applications and also the relevant statistical methods we'll use in those applications. Okay, so for the prerequisites, I would say some maturity about probability and statistics will be required because, for example, we will talk about maximum likelihood estimation as the starting point. So we will not go over like why we want to do maximum likelihood estimation to cover those basics, which will be covered in these two courses, which are the course numbers at UCLA. And so if you didn't take those classes, I would strongly suggest that you can take, there's a class I think offered at Harvard on their website EDX called the stats, or it's either stats or probability. You can check it out, 101. So that class covers a lot of the basics about probability and statistics. And I think they give very good intuitive examples there. So you can check it out on EDX and you can watch some course videos. You don't have to go through everything, but do watch the relevant ones. So, and also in my class, if you find anything unfamiliar to you, you can raise your hand and I can point you to the correct review material so you can make up the background. Okay, so there will be no textbooks, but I will post the lecture notes on CCLE. And also I will post the reference papers to CCLE. So regarding, this is a PhD graduate level class. So the course grade will be composed of homework assignments and the final project. So I think we will only give you 3 bi-weekly homeworks throughout this quarter. And we will post it to CCLE and you will submit your electronic copy of the homework to CCLE. And in general, we will not accept late homeworks, but if you have some special scenarios or situations during the pandemic, you can let me know beforehand. And we will use R programming. That's a recommendation, but if you use say MATLAB or Python, it's still okay. We don't really require you to stick with R. And for the final project, we will assign you a data set for everybody. in the 4th week. So the requirement is that I want every group with no more than 2 students. So 2 students can form a group, or if you prefer, you can work by yourself. Your own, just one person as a group, that's also allowed. So every group will formulate the question based on the data set. So the requirement is that you have to use the data set we provide in some way, but you can also add your own data set as a supplement, right, to give you more input. That's also allowed. So it's very open. And the question can either be a biological question or a statistical question, or both, depending on your background and expertise. Then each group will answer the question by using, but not limited to the data, to the statistical method we covered in this class, and not limited to the data set we provided. So you can come to my office hours to discuss your project ideas, and I will encourage everybody to start early on this project. And in the end, every group will write a paper with no more than 6 pages. I will provide a template. So then you'll be able to use that. So I will provide a LaTeX template, but if you prefer to use Word, please feel free to do so, including figures, tables, or references, all included no more than 6 pages for the final paper. And for the font size, 12 points, at least, single page space. And so in the 10th week, we will try to do a 10-minute oral presentation so that you can briefly go over your ideas in this class, in the class. But make sure we will try to decide the time, but after we see how many groups we have in the end, because we want to give everybody a chance to speak. So, and the paper will be due after the final week. So I think maybe probably this date, it needs a little bit checked. So maybe it's not Sunday anymore, but I'll double check this date. Okay, so basically for the course grade will be based on the homework, oral presentation, the paper. And I want to assure everybody not to worry too much about the grades, because this is a graduate level class. And so what you learn from this class is most important. And also for the project, I hope that everybody can find some connection with your research interest so that it can be useful. Okay, so these are the topics we will cover in the 10 weeks. Okay, so you can take a quick look. I may add some new results based on what I know, right? So basically, I try to update the contents year by year by adding some more cutting edge topics. And regarding the policy, so I hope most of you can turn on your video so I can see you, and we'll decide whether we need to move back. We can move back to in-person teaching after week 2. So we'll just follow the UCLA policy. And also, we will just use the CCLE as the only course website here for posting the content for doing your homework submission. So we will not move to the new platform for this quarter. So make sure that you can access the CCLE. And also, please keep your current email address with my UCLA. So if you didn't receive my announcement sent on Sunday or Saturday, then there might be some issue with your email. Then please check that out. So all the email conversations will be sent from my UCLA or CCLA. So that's all. Do we have any questions regarding the course policy? Okay, sounds good. And so just to let everybody know, if you have access the CCLE, let me try to do the screen sharing here. So for today, I think for today, it's an easy start, right? We just our first meeting. So I posted some papers to read under week 1. So for today, I think my plan is very simple. I just want to go over some key points I see in those papers. I think they are very general and they should be of broad interest. And then you can take your time to read the details in those papers afterwards, or if you have done so already, that's great. So, but my goal is just to go over the key points in those papers. And I think probably a question I have for you today, for everybody to think about, is what's the difference between what we call the statistical inference and or the machine learning, supervised learning, unsupervised learning? That's a question I hope everybody can get a vague or big high-level answer after today's class. And I will try to give you my interpretation, my interpretation on this question at the end of this class. But we can bear this question in mind throughout this class. And we have a question in the chat box. Is this class hybrid recorded? Yeah, I would still do the recording and post the recording regardless of we switch back to the in-person teaching. Yeah. Okay, so if you haven't done so, maybe you can download those papers from the CCLE for now, and we'll start from there. And also, actually, maybe before we start, I think in my previous lectures, I always ask the students around to just introduce a little bit about yourself and say why you want to take this class. Maybe we can, yeah, go back to that virtual just to give everybody a little time to introduce yourself. But I think the key is that you can let me know if you have a clear idea what specifically you want to learn out of this class, given your other classes you have taken or you want to take. I think that will give me a better idea about your background. So maybe we can start from Aditya, who asked the question.
Yeah, sure. Hi, everyone. My name is Aditya. I'm a first-year bioinformatics PhD student, currently rotating with Roel Ophoff in the psychiatry department. And I'm interested in taking this class because a lot of the kind of work that I plan to do in the future revolves around a lot of kind of robust statistical modeling. And I think it's important to have the fundamentals down before kind of getting into the weeds of the other things. So yeah, excited to take this class. Thanks.
And Charlotte.
Hi, everyone. My name is Charlotte. I'm a first-year bioinformatics student, and I'm working at Matteo Pellegrini's lab. The reason why I'm taking this class is because I realized I've been having a hard time to integrate statistics models with machine learning models. So I'm willing to learn more about it and how to analyze data using statistical model and machine learning at the same time. Yeah. Okay, thank you.
That sounds good. All right, Chris.
Hi, yeah, my name is Chris. I'm a second year statistics student, and I'm currently in Jessica's group, and I'm interested in taking this class because looking at the syllabus, there are a lot of methods that are related to my field in bioinformatics. So that's why I'm here.
Yeah, thanks, Chris. And next, I think Tim.
Oh, cool. So I'm Tim. I'm also a first year bioinformatics PhD student working under Eric Deeds on single cell RNA sequencing methods development. I want to take this class partly because I want to improve my foundation in the sort of statistical methods that people use in bioinformatics a lot. And this class seems to like dovetail very nicely with my area of research. So yeah, I'm excited for this class.
Thank you. Next, Christy.
Hi, I am a second statistics student in Jessica's lab as well. I wanted to take this class because a lot of the topics on there I have heard about in a lot of lab meetings, but maybe don't know as well as I probably should. So I'm hoping to get a better understanding of those things.
Great. And next, let me see, Guanghao.
Hi, everyone. My name is Guan Nao, and I'm the second year PhD student in statistics and also working in the Jessica's group. And I take this course because I think there are lots of statistical techniques I want to learn to integrate the statistics and the bioinformatics. And yeah, nice to meet you guys.
Yeah, thanks, Guanghao. And also, I think this is a good chance for people to briefly know each other's background. So then when you decide to form final project groups, you may find some, I would say, complementary expertise from the class, right? Then you can combine your expertise. And next one, I don't know how to pronounce the first name exactly. Gunalan. Maybe you can tell me how to pronounce it. Hi.
My name is Gunalan Natason. Yeah, hopefully it's just pronounced Gunalan if you're worried about the pronunciation. But yeah, thanks for having me. I am a first year master's student in the bioinformatics program, currently working with the Deeds lab. I have a prior experience in computational systems bio, and I'm sort of doing my thing in bioinformatics to branch out and get a little bit more experience. in other fields that I'm not usually accustomed to, which is also sort of my motivation for taking this course. Statistics is rapidly growing in the context of biology, and I sort of want to understand it before I actually start engaging in it.
Great, thanks. And next, Helena.
Hi, my name's Helena, and I'm a first-year bioinformatics PhD in Dr. Butro's lab. And I'm taking this course because I want to learn more about statistics and be confident about using it in my research.
Great. Your virtual background actually is the real background, right? You are using the real background. It looks like a very nice virtual background.
It's a basement.
I see. Okay, next one, Jack.
Hi, everyone. I'm Jack. I'm first year bioinformatics PhD, rotating currently in Dan Geschwin's lab. And I'm taking this class because a lot of my background is more on the computational and biological side of things. So I want to be able to answer those questions in a more statistical context.
Sounds great.
Thank you.
Next one, Grace.
Hi, I'm Gian. I'm also a first year bioinformatics PhD student, and I'm taking this class because I wanted to improve my knowledge in statistics and build a solid foundation. Thank you.
Thank you. And next, Jin Yo.
Yeah, hi everyone. I'm Jin Yo. I'm a first year computer science PhD student working with Harold Pimentel. I've been working on some projects involving statistical inference, but like most of the time I feel like I don't have enough understanding of statistics to actually compare the methods and like improve the methods. So yeah, I really want to take this class and I'm looking forward to it. Thank you.
Thank you. Next, Lillian.
Hi, my name's Lillian. I'm a second year MPH student in the biostatistics department. I'm interested in taking this class because I've heard a lot of the techniques and want to challenge myself and also better understand a lot of the statistical methods frequently used. So I don't have a lab, and so I might be less experienced than other people in this class, but I'm willing to challenge myself.
Thank you. And next, Maria.
Hi, everyone. My name is Maria. I'm also a first year PhD student in the bioinformatics program, and I'm currently rotating in Pivey's lab. I wanted to take the class just kind of the same reasons everyone else. I don't have too much statistical background, and I wanted to get more knowledge and experience to apply it in the field that I'm familiar with. I work in forensic genetics, and there's a lot of improvement to be made in terms of statistics in that part of the field. So that's why I'm taking this class.
That sounds exciting. Next one, Michael.
Hi, I'm Michael. I'm a first year bioinformatics PhD student rotating in Dr. Eric Deed's lab. I'm taking this course because, like everyone else, I want to better integrate statistics into my research and hopefully incorporate it in my future career.
Sounds good. And next one, Kathy.
Hi, everyone. I am a second year bioinformatics PhD student in Brent lab. The reason why I'm taking this class is because, like most of you guys, I have some basic high-level understanding of a lot of statistical concepts, but I would like to improve my foundation and also learn more to have a better understanding and how to apply the statistical methods.
Thank you. Sorry, I missed the lab name. Whose lab are you in?
Brent Fogel.
Oh, sorry, I didn't know him.
He's bioinformatics and also neurology, neuroscience.
Got it. Thank you.
Thank you.
Okay, and next one, Qingyang.
Hello everyone, my name is Qingyang. I'm also I'm a first year statistics PhD student, and I'm also in Jessica's lab. And I want to take this class is because I don't really have much experience with biological data and analyzing biological data. So I want to learn more learn more statistical topic using biological data and then how to use various methods to do the analyze. Yeah.
Great. So next one, let me see, Rochana.
Hi everyone, I'm Rachna. I'm a first year master's student in bioinformatics and I'm in Dr. Thomas Graber's lab. I'm also taking this class to improve my understanding of statistics and also to improve the statistical analysis of the research we're currently doing in our lab.
Great. Next student is Rosetta.
Hi, everyone. I'm actually a fifth year student in molecular physiology. So, and in Karen Roy's lab, and my background is actually wet lab, but I've been taking statistics and computational classes to improve my data analysis skills, as well as be able to kind of find that joint area between wet lab and dry lab where I can kind of like improve my research, and I just see how much potential there is when you're able to do that. So I want to learn more of that.
Sounds great. And the next one is Rio. Is that the right way to pronounce it, or Rio?
Rio.
Oh,
Yeah.
Rio. Yeah.
Yeah. Hi everyone. I'm Rio. I'm also a first year bioinformatics PhD student. I'm rotating with Noah Zeglin this quarter. I also, just like everyone else, want to improve my statistics background and apply it to my research.
Great. Welcome. Next one, Sandra.
Hi guys, I'm Sandra. I'm a first year bioinformatics PhD student and in Bogdan's lab. And the same reason as everyone else, I just want to improve my understanding of statistics and how I can apply that to various biological concepts.
Great. Welcome. And next one, Sandy.
Hi, I'm Sandy. I'm also a first year bioinformatics PhD student. I'm taking this class because I feel like my statistics background is a little weak. So I'm excited to build a strong foundation. And I work in Harold Pimentel's lab.
I see. Welcome. Okay, next one, is it correct to pronounce it Santiago?
Hi, yeah, it's Santiago. Yeah, so I'm a third year chem PhD student, and I'm trying to take this class because looking at the syllabus, I've sort of taken classes or done projects around some of the topics in this course. So I feel like I just want to fill in some gaps in statistics and my knowledge.
Sounds great. Okay, next one, Surabi.
Hi, I'm Surbi. I'm a second year master's in statistics student, and I'm working currently with Dr. McKinnon on my thesis about climate research. I'm also just interested in how statistics can be used towards biological problems. So I want to learn more statistical skills and learn more about that.
Sounds great. Okay, next one, Terrence.
Hey everyone, I'm Terrence. I'm a first year bioinformatics PhD student rotating with Noah Zetlin right now. And I'm, like everyone else, interested in expanding my statistical foundation, also because I'm broadly interested in pursuing statistical genomics.
Great. Okay, next one, Thomas.
Hi, everyone. My name is Thomas. I'm a second year toxicology PhD student, and I work for Xia Yang. And I'm in this class because my research is a combination of both wet lab and dry lab stuff. And my background is pretty much exclusively wet lab. And so to kind of improve the quality of my dry lab work and to participate better in discussions, project design, all that good stuff, I would like to improve some of the skills in statistics and get that understanding from this course.
Great. Next one, okay. I think Tim already spoke, and Wenxi.
Hi everyone, I'm Wenxi. I'm actually an undergrad in UCLA majoring in computational biology, and I'm working with Dr. Matilde Pellegrini. I want to take this class because I'm working on a project with him, and I think the statistical models can provide me with some inspirations from this class.
Okay.
Thank you.
Next one is Yu Fan.
Hi everyone, I'm Yufan. You can also call me Ivan. I'm a first year PhD student in Department of Epidemiology with Dr. Ritz. So my research interest is focusing on Parkinson's disease and all kinds of neurodegenerative disease. And also I think this course is very helpful and very related to some analysis based on the GWAS and also the metabolomics, also genomics. So I think it will be very helpful to take this class to increase my statistical background. So yeah, that's it. Thank you.
Thank you. And next one, Zhao Dong.
Hi everyone, my name is Zhao Dong and I'm a second year master student in biostatistics department. The reason I want to take this class is because I want to improve my statistical skills and learn some methods in the computational biology and the bioinformatics background. So yeah, that's it. Thank you.
Thank you. Okay, Zhi Qian.
Hello everyone, I'm Zhi Qian. I'm a first year statistics PhD student and I'm working in Jessica's lab. I didn't take any course related to bioinformatics before, so I hope I could learn how to combine statistical methods with biology and how to deal with the common issues of biological data types. Thank you.
Thank you. And our last student here is Yu Hang.
Hi everyone, I'm Yu Hang, a first year master student from the biostat department. And I used to do a lot of research on the biology, specifically in immunology and gene expression. I'm so excited to see like there is a course actually use the statistical methods on biological data. So that's why I'm here taking this class.
Great. Okay, thank you everyone for your self-introduction. We have a very diverse group of students here, so I'm really looking forward that you guys can work together and think we will have a very stimulating and diverse presentation in the final week. So, and let me introduce our special reader, Dongyuan Song here. So he is my PhD student and he is in the third year, right, bioinformatics program, PhD program. So in the same program as most of you guys here. So Dongyuan, would you like to say a little introduction about yourself as well?
Yeah. Hi everyone. My name is Dongyuan. I'm a third year bioinformatics PhD student working in Jessica's lab, and I will be your special reader for this class. I'm very happy to meet you all. So if you have any questions about the problems in class or questions in homework or just about the bioinformatics PhD program, you can always contact me. Thanks.
Sounds great. Yeah, and also I want to give everybody a little bit like an encouragement regarding the final project. So previously we had successful examples where some students can actually turn the final project into their first paper publication. So that's something it could happen if you really think it's something of your interest and you spend time into it. So that's why I feel like we try to give you some important, I would say interesting and important data sets from recent publications in the fourth week, so people can all work on the same data. And also because of that, I think the final projects will also be interesting because you can see how other people analyze the same data. All right, so thank you for the introduction. And also I would say we want to use the CCLE discussion forum effectively. So like if you have a question, it could be a general question you have from your research, or it could be a specific question about course material. You can post it to the discussion forum and we'll reply there. So then other people can also see the same question. So I think that can make the class more engaging. So for now, we will start with the review papers I posted on CCLE. And let me start sharing my screen. I think I will start with this very nicely written paper first. Let me see. And you can also have it offline by downloading it. So this is called A Guide of a guide to machine learning for biologists. I came across this paper last year during the pandemic. It was published in a Nature Review journal, I think molecular cell biology. So it was indeed written for biologists. And I think it gives a pretty good overview about the machine learning methods. So I will not read the text line by line because I think that would be too boring, but I think I will try to go over some of the points here. So basically for machine learning, right, bear in mind the question I have for you in the beginning, what's the difference between statistical inference and machine learning? This paper covers machine learning. And here, I think the major division of machine learning is the supervised learning and unsupervised learning. So this is the two big classes of methods, which we will actually cover mostly in the unsupervised part in this class. Okay, so for supervised, by the name, it suggests that we actually have some labels of the data, so for which we can predict. So for example, like the prediction of genome assembly to genome accessibility to genome regulatory factors. So this is the one application, right? So here, the thing we want to predict is whether some protein binds to this region, right? Given a specific region, we want to predict whether a protein binds it or not. So it's like a binary prediction, but we have some data available from previous measurements that tell us, yes, this protein binds here and here and here, and doesn't bind here, here, and here. So that will give us the so-called training data from which we can use the training labels to supervise us to build an algorithm and apply the algorithm to unlabeled data and predict labels. That's called supervised learning. And in contrast, the unsupervised learning means that we don't have any labels. We just have the data we call features, measurements. And based on the features, what can we say? So if we go down here, I think this is a very nice summary in this paper. So for example, among here, the five classical methods the authors listed, the D, PCA, and E clustering. So this is called dimension reduction. This is called clustering. They are unsupervised. So here the dots, each dot is one data point. So we can say one data point, or we can say one instance, that's the machine learning language, or we can say one observation, that's a more statistical language. And every point is represented by a dot. And here we have two features, height and weight, right? Or gene 1, gene 2, there are two features. So we're just using the features to extract information. So for example, what we can do is that here we find that weight and height are highly correlated. So probably we can combine them into just one variable, and we may not need two variables. And how to find one variable? PCA can give is a way. So here the PC1 is the direction in which most of the variance can be explained. So if we see the PC1 alone can explain, say, 80%, 90% of the total variance of the two features together, then we may just be happy with PC1 as the feature alone. So this is just a very nice illustration of PCA, and we will talk about the detail in our lectures. And clustering is another data exploration. So given the features, we have the data points, and intuitively we may see the data points fall into clusters. Then how can we identify the clusters? So you can see that here there are no labels involved at all. We're just trying to cluster data points based on features alone. Okay, but if you look at here, regression, support vector machine, gradient boosting. So these three are supervised learning methods or algorithms. So they would require labels or some response. So for example, here, let me just say that here the data points are labeled as ordered proteins or disordered proteins. So colored by two different colors. And the task here is to find a separation hyperlane. So here we are in the two-dimensional space. So then the hyperlane becomes a line. How can we find this line to best separate the two classes in the hope that if we have another data point without a label, we can put it in the space and use the hyperlane to decide whether we should call it ordered or disordered. So that's the supervised task. So you have the labels beforehand in contrast to clustering. And what's the difference between, say, regression versus the B and C? So here it's just depending on whether the label or the variable we want to predict is continuous or is discrete. So if it's continuous, we call the regression problem. Here, like say, we want to treat the weight as the variable for prediction, and height is the feature as our input. So output is weight, input is height, and weight is continuous. So we treat this as a regression problem. And here it's just a linear, simple linear regression, right? The prediction is also continuous. And versus here, when we say the labels are discrete or categorical, we call this a classification problem. So here we have two classes, blue, red, representing order proteins and disorder proteins. And here the gradient boosting, right? We're trying to predict the drug, I would say probably the drug status active or inactive. So basically here the prediction outcome is not continuous, it's discrete, and we call this a classification problem. So I want to draw your attention to all the 5 figures together. So if you think about machine learning, and if you think about it as a decision process, we're trying to make decisions, then what is our target? What do we make decisions for? I can pause here for maybe 10 seconds. So if you look at all the problems, essentially, we are basically trying to make decisions for our data points, right? So like here, we want to know which cluster each data point belongs to. And here, we want to find a way to represent our data, the whole data set, all the data points, using fewer dimensions, maybe not 2, but 1. And here, we want to predict the response variable for each data point. So I would say, based on my understanding, all the machine learning techniques, we are concerning data points. And data points are our, I would say, central role players here, right? So our decisions are concerning them, right? Whether the data points are labeled or not, it's just a matter of fact, data availability. But what we are concerning, our decisions are about, are the data points. So later you will see how this is very different from statistical inference. But this paper alone only covers machine learning. Okay, and figure 3 is about traditional machine learning. I think by traditional, it means that it doesn't use neural network. And then another figure here is about those common, typical neural networks that have been used in biomedical research, bioinformatics research. Like this is like, you know, this is just a fully connected neural network. And this is convolutional neural network, which means that you have this convolutional filter layer to just get this nearby pixels to extract information from there before you build a neural network. And there's a recurrent neural network that applies to like the data structure where you have this kind of transition chain, like you have the DNA sequence, right? A nearby C, nearby T, nearby C. Then you put that, you know, the recurrent structure into your neural network building. Or you have this graph convolutional neural network where here your data is a graph instead of an image. In the image here, you consider the convolution is about nearby pixels, while here is about nearby nodes connected by edges. And also the autoencoder is more like an unsupervised version of neural network where your goal is you don't have labels, but you have the input data here and your output is something you want to mimic the input in the hope that you can find some low dimensional latent representation. You're going to use it for some downstream analysis like the clustering. So out of those, I would say A, B, C, and D are supervised neural network. E is unsupervised neural network. So this is just what this paper tries to summarize, right? For the neural network method, I would say in the most simplest language to say what it does is that try to make this function here, the linear model here, or the hyperplane here, more complicated. So, and the more complicated function is represented by the neural network. But still, the nature is the same. It's either supervised or unsupervised. And the difference is the complexity of the function you try to estimate from the data. Okay, so I think for this paper, it's very readable. And also this table here is very nice. It just gives you for a different input data, what tasks can be done and what are the recommended models. Maybe not, people may have different opinions, right? This is not like the gold standard, but this is the author's recommendation and what challenges are there. So you can see in bioinformatics, the input data can be structured tabular data like gene expression. So for gene expression data, and this is the focus of this class because it's the simplest data structure. It's already in a matrix format. Like when we say gene expression data from patients, then we usually would have, say, the columns as genes, which are usually regarded as features, and the rows as patients. So every patient is regarded as one observation. So that's one matrix. And if we talk about single cell gene expression data, then we may have corresponding columns as genes, rows as cells, but some people may just put the matrix in the transpose way. So rows are cells, rows are genes, and columns are cells, but it doesn't matter that much. So at least we know that either the rows or columns correspond to variables and the other dimension will correspond to observations or data points. So gene expression data will be our focus. And on gene expression data, we could do unsupervised learning like clusters, right? We could either cluster cells, patients for this dimension, or we may regard genes as data points. And when we can cluster genes, that's also possible. And we can also do dimension reduction so that we can represent each patient sample or each cell as a two-dimensional data point for visualization. And that's what PCA can do, or more complicated dimension reduction methods like t-SNE or UMAP can do. So these are very related to what we will cover in the class, but I want to say that this table lists other input data that are less simple, like gene sequence. These will be ATCG nucleotide sequences or protein sequences composed of 20 amino acids, or even protein three-dimensional structure, or mass spectrometry data that's more like signal tracks, or images, like these are from medical data, like brain imaging, and also molecular structure, like chemical structure, right? Molecular structure, and or protein-protein networks. So in those cases, the input data can be called, say, raw or unstructured, which means that they are not numbers and they are not in matrix. So in this case, we need a lot of pre-processing to convert the data into some numerical format so we can work with. But I would say these are not really the topics of this class. So we will focus on tabular data throughout this class, and because that's easy. And also that's what statistical modeling can be the starting point. Otherwise, for other raw data, we need a lot of pre-process steps before we can talk about statistics. Okay, so this is what I think I want to say for this article, and also this box too, regarding those topics or considerations we need for doing machine learning is actually very good. So I think I would suggest everybody to quickly go through this box and so you can get a better sense about machine learning. Okay, but actually I want to say that compared to statistical inference, machine learning is actually more straightforward to understand. Especially, I think this figure gives a very intuitive summary about what's supervised learning and what's unsupervised learning. So the next paper I want to talk about, also I put in my folder, is the paper I wrote in the year 2020 during the pandemic. So this is the paper I wrote about the distinction. So I think this can be the starting point for our discussion: statistical inference versus machine learning prediction. So in here, I talk about the specific task regarding hypothesis testing versus binary classification. So actually, if you learn about those two things from your undergraduate classes or some other classes, usually they are taught in different classes. Like you may learn hypothesis testing from a statistics class, like the Stats 100B I'm teaching right now. And you may learn the binary classification from another class that teach you machine learning, right? Like the examples we saw before, SVM, gradient boosting, right? These are about binary classification. So how can they be confused? Maybe I can quickly go to the example. Actually, it's a kind of a motivating example here. And actually, we can start from here and then discuss this topic. Okay, so this is a problem called cancer driver gene prediction. And here the goal is to utilize an individual gene's mutational signatures, such as number of missense mutations, to predict how likely the gene drives cancer. So for many genes, we don't know whether they drive cancer or not. And that's the goal for prediction. Note that the mutational signatures are aggregated from multiple patient databases, and individual patients' data are unavailable. So therefore, for every gene, we have some signatures built from a lot of data from patients, but we don't have individual patients' data. We have knowledge of a small set of cancer driver genes and neutral genes that are unlikely to drive cancer. So basically, we have a small set of cancer driver genes, and we have a small set of neutral genes. So you can consider neutral genes as non-cancer driver genes. The question is, how can we leverage this knowledge to predict whether a less studied gene is a cancer driver gene? So this is the data analytical question, how to do the prediction. Okay, so based on what I just said, what's the intuition you have? How do we formulate this problem? So given that we know some genes are cancer driver genes, some genes are neutral genes, but for the majority of genes, we don't know, and we want to make prediction. So to me, this is like a supervised learning problem, right? So we want to learn a classifier from the known cancer and neutral genes so we can predict for new genes. But when I was asked this problem, I was surprised to learn that a famous algorithm called Tucson addresses this question using not a classification approach, but a hypothesis testing approach. So in brief, it regards mutational signatures as features and uses hypothesis testing to assess how much an individual gene resembles known neutral genes based on each feature. So let's say we have 5 features, then for each gene, we are going to assign it 5 p-values. So by doing 5 tests. So basically the strategy is that the gene's feature value is used as the test statistic whose distribution under the null hypothesis, that is the gene is a neutral gene, is estimated from the feature values of known neutral genes. Then from the test statistic and the approximate null distribution, the gene receives a p-value for that feature. Suppose there are 10 features in total, then each gene receives 10 p-values, which are subsequently combined into a single p-value by Fisher's method. Okay, so basically, let me just, it's not in this illustration, but this is how it works. So in other words, it will give every unknown gene a p-value by combining 10 p-values obtained in this way. So for example, if the neutral gene has low values for a feature, and this unknown gene has a high value for that feature, then this paper, this Tucson paper will assign the right tail probability for the unknown gene's value into a p-value. And it does so for all the 10 features, obtaining 10 p-values and combine the 10 p-values into 1 p-value. And here's my argument about why I think this is not idealistic. So from a statistical perspective, there are 4 apparent issues with this hypothesis testing approach. First, each hypothesis test 1 per gene per feature only utilizes the known neutral genes to estimate the null distribution, but does not fully capture the valuable information in known cancer driver genes. So this set is not touched. To be exact, Known cancer driver genes are used to select the predictive features before hypothesis testing is performed. However, in each test for one gene and one feature, the information of known cancer driver genes is only partially reflected in the direction of the gene's p-value, two-sided, larger than, or smaller than, which excludes the possibility that the known neutral genes may have feature values on the two sides of those known cancer driver genes. So in other words, here the cancer driver genes are used in a very indirect way just to decide the direction of the p-values. Second, each hypothesis test is performed using a sample of size 1. That is, the test statistic is the feature value of one gene, which is known to be not powerful and thus undesirable. That is, if the gene is a cancer driver gene, we may miss it with a high chance. This is the reason why we recommend using more than one instance, but that is sample size greater than 1, for hypothesis testing. And I'll go back to the table 2 there. And third, combining multiple p-values into a single p-value is a difficult task, especially when p-values are not independent of each other. The fact that mutational signatures are observed to be correlated features, their resulting p-values are correlated for each gene, violating the independence assumption of Fisher's method. Although there are methods for combining dependent p-values, they cannot address the most fundamental question: what is the population behind each hypothesis test? Leading to the last issue. Fourth, the population behind the null hypothesis is unspecified. For a given test, is the population about that particular gene or all genes? Therefore, we conclude that the hypothesis testing approach is inappropriate for this cancer driver gene problem, which instead should be formulated as a binary classification test by the reasoning below. Okay, so I think this is just like, maybe I should go back to the guidelines here. So I think the key difference between testing and classification is regarding whether the questions are about the instances or data points or the features. So here I'm just making this hypothetical example, not a cancer driver gene prediction problem, but a new problem regarding this matrix being rows as patients and columns as features, as genes. So we regard genes as features, patients as instances. So here, every entry in the matrix reflects the expression value of a gene in the patient. So here, let me say I label the patients previously as diseased, yes or no, or unknown, right? So let's say I have these patients as not diseased, these patients as diseased, but for this first row, this patient number 1, I don't know whether it has a disease or not. So if the question is about is patient 1 diseased, then this is a classification problem because it's a problem about an instance. So what we do is that we use the training data with labels and an algorithm. For example, the algorithm could be a neural network, or it could be just an SVM, or even logistic regression. Then together, I build a classifier. And then I apply this classifier to the instance, right? The instance, the first row is my input, and I will get a prediction. So the prediction might be no. So that's my answer. And if I'm interested in the gene instead, not a patient, right? So if I have a gene A, and I'm interested in knowing whether this gene is a biomarker. By biomarker, I mean, can this gene's expression level really differentiate the diseased patients and the undiseased patients? So in this case, I'm going to take all the patients with labels together as my instances, and I do a statistical test. And I will decide whether this gene can really distinguish those two groups. And if the result is reject, the null, I will conclude that the gene is a biomarker. So now the question for you guys is that, can you see the difference between these two approaches, the binary classification and the hypothesis testing? So I think the most obvious distinction is that for the hypothesis testing, my binary decision, reject or do not reject, is based on all the instances together. While for the binary classification, my prediction decision is for one instance only. So if you think about the decision rule, what is the rule? In binary classification, the rule is a classifier. And the rule is trained, it could be trained by some training data, right? But you can imagine the training data is not always necessary. If you are smart enough, you come up with some good classifier without using any data. It might work well based on your knowledge, right? But that's still a classifier. So the classifier is the rule that gives you the output, give you the decision. While here, the statistical test, it's also a rule. But does this rule require any training? Actually, no. The rule is just applied to all the instances together, and it will give you a binary decision. So therefore, you see, I think in this example, you can see the distinction is that for hypothesis testing, our conclusion is based on the whole sample, but it's really about the underlying population. The population is the truth, right? Is this gene a biomarker or not? While in the binary classification, or more in general, supervised learning, it's about making decisions for individual instances. It's about that particular instance. It's not really about the population. So I hope this makes a little bit sense to you. And also just go back to here, this table. Probably this one is actually even more clearer. So I think I might introduce the 4 concepts here under each framework. And so I would say I come up with this table, but you might have some better ideas about how to formulate this. But I think I divide the concepts into what's the binary question, what's the binary answer, what's the decision rule, what's the binary decision. Okay, so the binary question for hypothesis testing is actually, is the null hypothesis false? Actually, unfortunately, this question is not answerable because we do not see the whole population. We do not see the truth. We only have a sample from it subject to randomness. So this question, even though it's the fundamental question, but we don't know actually. And for binary classification, it's actually, does the instance have a label 1 or 0? Okay, so the answer is binary to each question. For the hypothesis testing, if the answer is no, it means that if the answer is no, it means that the null hypothesis is true, and 1 means the null hypothesis is false. Unfortunately, these answers are still unobservable. Well, for binary classification, the binary answer is the 0 label, 1 label, and this is observable. Okay, and for the decision rule, the hypothesis is that a statistical test that inputs data and outputs a p-value, which is compared against a user-specified significance level alpha. If the p-value is less or equal than alpha, our decision is rejection, reject the null. Otherwise, our decisions do not reject the null. Okay, under classification, the decision rule is a trained classifier that inputs an unlabeled instance's feature values and outputs a predicted label. And the label is either 0 or 1. So therefore, you can see that, and in those 2 cases, the binary classifications question is concerning 1 instance, while the hypothesis testing is concerning the fundamental truth, what's in the underlying population. And even though we make a binary decision, right? But here, this decision is depending on our sample. It's not necessarily the truth. That's why when we do statistical hypothesis testing, we may occur a type 1 error or a type 2 error. And that's when that happens when our decision is not the truth. Okay, so seeing these 4 concepts, now let's move to here. So there's a side-by-side comparison between the 2 formulations. So first of all, is it symmetric? So for hypothesis testing, it's actually asymmetric, which means that the null hypothesis is the default, and we stay there unless our data give us a very strong evidence to reject the null. So in other words, when we do hypothesis testing, we're taking a very conservative perspective. The null is the default, and we do not easily reject it. Well, for binary classification, in most common test is symmetric. Like we don't think the mistake of calling a 0 as 1 and the mistake calling 1 as 0 have unequal weights. We treat them equally in the loss function. However, more recently, there are asymmetric classification, and I have some research in that aspect. So we could actually give the two types of errors different weights. Okay, and now the number of instances we use to make a decision. As we have seen, usually for hypothesis testing, we require a large sample size, and the larger, the better. And also statistical theory tells us if a test is valid, then if your sample size goes to infinity, the test power should reach 1, should converge to 1. That's what we call valid test. But for binary classification, we make one decision per instance, right? We will do a prediction for every instance. So that's different. And so regarding the availability of binary answers, for hypothesis testing, no, because we said we don't know the truth. We are not a God. But for the binary classification, if we have training data, then yes, we regard the training data labels as the available binary answers. And regarding the evaluation criteria, so for hypothesis testing, we use power. The more powerful the test, the better. And so the power are compared under a given significance level, say 0.5. Under the same level, we compare different tests, say t-test, Wilcoxon test, and see which one is more powerful, right? Well, for binary classification, we talk about prediction accuracy. We could talk about the precision, the recall, and also we could talk about the false positive rate, false negative rate, those kind of criteria I listed in this article. These are used for binary classification. And the most striking comparisons here, and this is more theoretical, it says that if we have the largest possible number of instances, let's say we see the whole population, and then the hypothesis testing will give us a power of 1. That's guaranteed. But the prediction accuracy is not necessarily perfect. So for this answer, let's go back here, right? So let's say if we have collected data for every person in the world, then measure the gene expression in every person, then we know for sure whether this gene A is a biomarker, right? Because then we see the truth, we see the whole picture. But still, even though you have every person measured in the world, with a new person, like a newborn baby, if you want to predict whether this person will have a disease or not, you may still not have 100% accuracy. The reason is that the diseased people and the undiseased people may share some overlap in the features. So there's always some ambiguity to make the prediction. So that's another contrast to see that hypothesis testing and binary classification are indeed different formulation. So now let me try to say that the guidelines I provided here, I think these are very useful, and also these are related to the question asked in the beginning. What's inference? What's the prediction? So guideline 1 is decide on instances and features. That's the key. So now given a table like here, it's not labeled as instances and features like here. It's just like a table of a matrix. Then as a data analyst, you need to decide which do we regard as the observations or instances, which do we regard as features, right? So this is the first question to ask. So let's revisit the guideline here. So given a tabular data set, the first and necessary step is to decide whether rows and columns should be considered as instances and features respectively, or vice versa. The decision may seem trivial to experienced data scientists when columns represent variables in different units, say gender, age, and body mass index, in which case columns should be considered as features for sure. However, the decision may become not so obvious in certain cases. For example, in figure 1, in a gene expression data set that has rows and columns corresponding to patients and genes, respectively, and all data values are in the same unit. The question is, should we consider patients as instances or features? To answer this question, the key is to understand instances as either repeated measurements in the data collection process or a random sample from a population. This is the key to understand instances. What do we consider as a sample? Gene expression data are collected to understand gene expression patterns in healthy and diseased human subpopulations. So healthy and diseased patients are considered two random samples, each from one subpopulation, and satisfy two. Hence, we conclude that patients are instances and genes are features. In general, the answer depends on the experimental design and the scientific question, both of which will determine what the underlying population is, as we see in the cancer driver prediction example. So this section. Because in this case, I'm regarding genes as instances instead of features. So I think it's depending on the context. And guideline 2, list the binary decision to be made. The second step is to outline the binary decisions to be made from the data. Formulate analytical tasks such as biomarker detection and disease diagnosis into binary questions for which binary decisions will be made. Divide binary questions into those related to features and others concerning instances. For example, whether a gene is a biomarker is a feature-related question, and whether a patient has a disease is an instance-related question. Hypothesis testing can only answer feature-related questions about the population, while binary classification can only address instance-related questions. Guideline 3, assess the availability of non-binary answers in data. After a list of binary questions at hand, the next question is, do the data contain any known answers? If we already have an answer to a binary question, we cannot formulate the question as a hypothesis testing task. In the case where some instances contain known binary labels, but we concern about the unknown labels of the rest of instances, we are facing a binary classification task, just as in disease diagnosis. Otherwise, if the data contain no binary labels, we do not have training data to construct classifier, which, if not given, will prohibit us from predicting unknown labels of instances. In guideline 4, count the number of instances for making each binary decision. Suppose that we are given a decision rule, such as a statistical test or a classifier in the form of a formula or a computer program that can take our data as input and output a binary answer. An easy check is to count the number of input instances needed to output each binary decision. If we're expecting one decision per input instance, it's likely a binary classification task. Otherwise, if each binary decision needs to be made from a group of instances together, the task cannot be binary classification, but might be formulated as hypothesis testing. And the guideline 5, I think this is the most fundamental one. If you know this, then probably you don't need the previous 4. The most fundamental check is to evaluate each binary question by its nature. Is the question regarding the unseen population of which our observed instances are a subset, or regarding a particular instance? Asking whether a gene is a disease biomarker is a question of the former type, as it concerns whether this gene can distinguish the human subpopulation carrying the disease from the rest of human population. In contrast, asking whether an individual has the disease is a question of the latter type. Hypothesis testing and binary classification are designed for answering questions of the former and latter type respectively, as shown in figure 1. So this is what I summarized regarding this binary decision scenario. So I will say that if you are interested in this, you can continue reading this paper, the details of this paper. But actually, this is based on my own experience about how challenging it can be to formulate a question into a statistical one or a machine learning one. I really don't want to place too heavy on their distinction. I want to say whether formula as a hypothesis testing or binary classification, it can be very confusing, especially when your data is of this tabular form. Okay, so that's all I want to say about this. Enough details. So regarding this, right, regarding the statistical hypothesis testing topic, there are some relevant articles I put in the folder. So one is this, called p-values are just the tip of the iceberg. So when we talk about p-values, we are doing hypothesis testing. And that's very common in our scientific investigation. So you see, we have to go through a lot of steps to obtain a p-value. So first, you need to design your experiment, collect your data, and then process your raw data, right? Clean it, and then explore your patterns in your data, and come up with potential statistical models, do the modeling, and obtain summary statistics from the model, and then you do inference. So here it comes to the word inference, right? What is inference? So inference by the term itself, it means guessing. We're trying to guess about the nature, about the population based on the data at hand. So here the summary statistics are just numbers we computed from the data at hand. But we're not just interested in the data per se, we are interested in the population from which the data are sampled. So that's why the inference and the p-value are essentially talking about, given the summary statistic, how certain we are going to trust our decisions, right? So roughly speaking, if the p-value is correct, then the smaller the p-value, the more confident we are about our rejection of the null hypothesis. So that's why many people prefer small p-values. However, this article is saying that those processes, those steps are so important that you cannot ignore them and just focusing on the p-value. Otherwise, the result could be completely misleading if you are doing the wrong formulation, wrong modeling, and wrong inference. So that's why I want to make people just have this, high level impression from today that when we talk about inference, it's usually very tricky. The reason is that we are making a guess, and we want to make sure our guess is reasonable with enough support, enough justification, not just a p-value. Okay, so, and also why I say machine learning, supervised learning is actually a little easier. It's because you have the training data with the labels for you to check, right? If you just leave out some of your training data untouched as your cross-validation, then that can give you a sense about whether you are predicting well enough or you're just overfitting the training data. You can know that. But for this hypothesis testing, as I said, there's no answer available. So the inference, like your p-value could be 0.001, but my p-value could be 0.2. So in this case, our conclusions could be totally opposite. Which one is more trustworthy? We don't have training data, so we have to just go through these procedures to justify each step and convince people. So that's why I never think that statistician, this job can be easily replaced by AI because we have so many steps get involved and we need a lot of subjective decisions in the middle. Okay, that's this paper. And related, there is another thing also written by the same group of authors I'm going to share. So that p-value iceberg is on Nature and this is on Science. So what's the question? So basically the author's message is that mistaking the type of question being considered is the most common error in data analysis. So we have to, this also echoes the importance of question formulation. So basically, this is a very nice diagram. It means that sometimes we may just be happy with descriptive statistics, like just report the mean, report the variance, or we may just, but we may want to go deeper, right? We may want to just do inferential, that's something I talked about, hypothesis testing, or prediction, more like supervised learning. And we may not be happy with here. We may even want to do causal inference, or we want to build mechanistic models like based on physical laws, like in systems biology. So we can go deeper and deeper in our data modeling. And this paper just gives you some examples about what's the question you want to ask. And depending on the question, we may want to go to different levels in our data modeling. So I strongly suggest that people try to read this paper. And I think in our first homework, I will ask you to give an example for each task based on your understanding. Okay, so I think we are about to running out of time. So I think this one, let me quickly go over the last two papers as an overview. So this one the pitfalls, 3 pitfalls to avoid in machine learning. So you may read this one together with the first paper. It's about machine learning, about that machine learning to biologists. So this one gives some very interesting examples where the machine learning may fail, like splitting data inappropriately, the existence of hidden variables, and mistaking the objective. Mistaking the objective is also aligned with that science article about the question formulation. And finally, this one. So this one is probably the most technical one. Maybe you will not fully understand it right now, but hopefully you will find it more understandable at the end of this class. So 10 simple rules for effective statistical practice. So these are written by a group of prominent statisticians based on their experience. So they give the rule one by one. So like statistical methods should enable data to answer scientific questions. Signals always come with noise. This is something we should bear in mind. Don't take our data as perfect. If it's perfect, there's no uncertainty, then we don't need statistics. And the third, plan ahead, ready ahead. This is about experimental design. Worry about data quality. This is about after obtaining your data, how do you check the data to make sure it's okay? And rule 5, statistical analysis is more than a set of computations. This is something we should bear in mind. We are always doing something, we should do something that's well justified. And as I said, it could be subjective. There's no simple set formula for doing the analysis as always. And keep it simple. I agree with this because simple ways of analysis can usually give people more transparent results. And rule 7, provide assessment of variability. This is a key. We always talk about uncertainty in our data analysis, as we will see again and again in our class. Check for your assumptions to make sure they're justifiable. When possible, replicate. This is about data generation. Having replicates is always better than not. Make your analysis reproducible. This is about like the responsibility in our paper, right? We need to make analysis really fully transparent, reproducible. Yeah, so that's all. I think I will stop here. I give you a lot of high-level ideas. And hopefully at this point, you have a better understanding about the question, statistical inference and machine learning. What are the distinctions? So if you still think this sort of misleading or mysterious or confusing, you please feel free to post your questions and thoughts to the discussion forum. So that's all for today. And we just give you high-level ideas today. And starting from next lecture Wednesday, we will talk about technical stuff. So we'll start with gene expression data analysis as our first topic in this class. So I think that's it. And I'll see you on Wednesday.
Thank you.
Thank you.
Thank you.
Thank you.
Bye. Bye-bye.